notice I posted on YouTube this morning, 12:30. By 7:30, class had to that in. I gave 5 exams yesterday. So I went to my son's basketball game Saturday night and was working on my computer all the way down there, going through exams, writing the exam and stuff. So I finally got them graded. Not quite sure everything was done right, but anyway, I got it done. You can double check whether it was done right or not. Anyway, and I didn't have time to, I have quizzes set up, but not the way we're doing it with the ABC used. That would have taken me another hour. And then I would have been too sick to come to school, so. We don't have anything due on Thursday, so we're taking today's quiz. Same material, that first page on the lab from Thursday, and all the parts of the heart, the valves, and the blood supply. But conduction system and histology won't be on. Okay, so we'll have what was supposed to be today's, just transfer it to Thursday. Okay? If we don't have additional, I don't like to have more than one thing to do on a particular day. Alrighty, and I'm still working on the homework. I'll get those back to you. Oxygenated blood is found in which of the following? So, where would we find it? B and C and E. Okay? So it returns, deoxygenated blood is coming via the superior vena cava into the right atrium and right ventricle, and then going to the lungs to pick up oxygen, and returning, remember the left side of the heart is our oxygenated side, so it's returning via pulmonary veins to the left atrium, and it comes out to the body via the left ventricle. So B, C, and E. All right, the anterior interventricular artery, and all of you can close your eyes and see right where it is right now, right? Okay, what's that a branch of, by the way? Coronary artery, but which one? Left. Left, good, okay. And it's going to be associated with which of these venous structures? Great cardiac vein. Okay, so it passes down the anterior aspect of the heart while the great cardiac vein is moving up and around the back of the heart. All right, cardiac muscle is most similar in appearance and structure to smooth muscle or skeletal muscle. Probably skeletal. It has some characteristics of smooth, primarily just the fact that it has one nucleus. All right, but everything else about cardiac muscle is going to be similar to skeletal muscle. We actually have 2 different types of muscle cells that we're going to talk about today. We have contractile cells, and that's what you're seeing up here. Those are the ones that are similar to skeletal muscle. And then what we're going to talk about first physiologically are the autorhythmic cells, the ones that actually create the electrical current that causes the contractile cells to contract. So when we draw a diagram of cardiac muscle cell in appearance, let me put it here. First of all, right off from the start, they look different from skeletal muscle because they are branching. And they form these irregular foldings where they join with another cell. And so then another cell would be coming here and joining with that. All right, we don't see branch cells in any of our other tissue types. Smooth muscle cell doesn't branch, but tissue types will be muscle cells. Skeletal muscle isn't branched, it's one long cylindrical structure. So there is a single nucleus, kind of boxy or rectangular in shape. And the mechanism or the structure that makes it look the most like skeletal muscles is the banding. So we have light and dark bands going perpendicular to the length of the cell that make it appear striated. And what causes the striation in skeletal muscle? what do we call the smallest unit of contraction? Sarcomere. And we have overlapping actin and myosin. All right, and we have that same structure in cardiac muscle. Smooth muscle has actin and myosin, but they don't have sarcomeres. They don't have the myofibrils. And so these striations are due to the A and the I band overlapping. Now, we have additional, if you look in the microscope, and I ask about striations, you can see these longitudinal bundles. Those are just myofibrils that are more noticeable as distinctive structures in cardiac muscle. Striations are the perpendicular banding, all right, that we refer to. And then you can see where the two cells have joined together. And with the additional folding of the membrane occurs, that makes it thicker. And so it's visible and identified as an intercalated, which means interdigitated, intercalated disc. All right, the intercalated discs have two major types of junctions. So if we take this and enlarge it, all right, we have here, we have gap junctions. Remember looking at gap junctions or studying gap junctions in Bio 430? They are made of 6 proteins that form a cylinder in each cell membrane, the connexons, and that creates small channels that are present between the cells. And this is important for allowing communication, because this is how we send a signal rapidly through the heart, resulting in the contraction of successive waves of muscle, right? Rather than having each cell having to wait to get innervated and controlling that pattern by nerves. On this area, we have desmosomes. So this is where the stress is going to be put as two muscle cells shorten and pull against each other. So we have that protein plaque strengthened by intermediate filaments. That's what we have holding all of our epithelial cells together. And these cardiac cells have those desmosomes as well. And that holds the cells together. All right, so the gap junctions are for communication. They also help to hold them together, but we need them for the communication. And the desmosomes are structural, actually holding them together. All right, so striation appearance similar to skeletal. A single nucleus is what smooth muscle has. What about innervation? Which are they most similar to? Cardiac muscle is most similar to smooth muscle, or skeletal muscle by innervation? Smooth, because it's autonomic. Okay? You don't decide. Biorhythms. Certain individuals with proper training can decrease their heart rate by just thinking about it, but they certainly can't stop it. All right? Like you could say, can you stop your breathing voluntarily? Can you say, I'm not going to breathe? Yeah, I think the maximum length of time is almost 8 minutes, or around 8 minutes of holding your breath. All right, so you couldn't make your heart stop for 8 minutes, or even 30 seconds. All right, so it's involuntary innervation. We'll talk about that a little bit more today. All right. Now, the cells that we're going to talk about, or these are just showing the junctions here, are the autorhythmic cells first. They were cardiac contractile cells that are modified. They don't have the actin and myosin. They are not striated. That's been replaced by glycogen as a fuel source. And so I want to do some review with you for nerve and muscle irritability. What allows a cell to be irritable? What do we mean by irritability? A characteristic that is found in only nerve cells and muscle cells. If you're irritable, you are, what do we like sensitive. Any little thing can set you off. So nerve and muscle cells respond to stimuli. All right. So as you recall, when you first started learning, in my class we did nerves first. All right. So however you did it, we first started learning about potentials. What did we call this potential? If we measured it by taking a cell and comparing the current inside the cell to the current outside the cell. And this would be, let me complete this. Okay, so I heard it, resting membrane potential. All right, so a membrane potential is the relationship between the positive ions just inside the cell membrane and just outside. So when we say that the resting membrane potential is a negative 70, let me move this picture over here. We mean that if we have a plasma membrane, there are more negative charges on the inside or fewer positive charges on the inside than in relationship to the outside. It's right at the membrane we're talking about. Okay? There are three conditions that provide this negative membrane potential. It varies between minus 55 and minus 70 for muscle cells and nerve cells. Anybody remember from Bio 430 what those three conditions are about the cell that provide the inside the cell being more negative in relationship to the outside?
Permeability.
Pardon?
Permeability.
Yes, that's exactly the word I heard this morning, but permeability of what?
Sodium and calcium.
Calcium will come into play with the heart, but it's not for the resting membrane potential. So permeability of what creates the permeability? Let me put it that way.
Channels.
Okay, channels. So let's stop there and let's go look at channels first. What kinds of channels do cell membranes have? Because this is something I want you to be able to apply to a diagram of skeletal or heart muscle cells. So what kind of channels?
Sodium.
Why do we need a channel, by the way? What's passing across the channel causing this? Ions. Ions are charged particles, and our plasma membrane is nonpolar covalent. It's a lipid, it's an oil. And charged particles aren't going to move very well across that. So we need to create a channel to allow the ions to move into or out of the cell. That's what makes it selectively permeable, is what actually goes through the channel. Okay? So we had just the lipid membrane, the ions aren't going to move through just the phospholipids. So we have to have this channel to allow that to occur. So we have channels that are open all the time. What do we call them? Leaky channels. All right, and then we have channels that aren't open all the time, and we call them gated channels. But different things will open the gates. So what three different things, major things, will open a gate? We're not going to consider electric currents here, but electrical channels. What's something that will open a gated channel and make it open? A ligand. Okay, a neurotransmitter. Some molecule will bind to that. So we have ligand-gated channels or chemical-gated channels. This could be a hormone or a neurotransmitter. What other kind of channel do we have? Voltage-gated channel. The next one's not very common, and I usually illustrate it during Bio 430 by asking students to close your eyes. How do we normally see? We have the energy of a photon affecting voltage channels, changing sodium channels, all right? But you can close your eyes and you can create light with your fingertips. Close your eyes gently. I've got soft contacts, but if you have hard contacts, you might want to be a little more careful. Close your eyes and just press on the surface of your eyeball. And you can make a light show, like a laser light show, on the back of your eyelids. What you're doing is putting pressure on your eyeball, and instead of the photon changing channels being open or closed, it's the pressure that you're putting on your eyeball doing that. And the brain that's responding to those impulses doesn't know where those impulses are coming from, and it creates light and projects it back to the things I do when I'm bored in meetings. And projects it back to the back of your eyelids, which are closed, so that's where you see it. All right? We also use it in ear. And sound waves are converted to vibrating bones, which are converted to fluid waves, and those open channels. So when we use force or pressure to open it or close a channel, what do we call that? Mechanical. Now, mechanical gated channels really aren't that functional. Well, not that. They aren't functional with our heart, but I just wanted to put a list of these up here. Okay, so we got an idea of what's going on. So our resting membrane potential is going to be is affected by permeability. How easily things move across the membrane into the cell or out of the cell. So what makes it a -70? What makes the inside of the cell more negative than the outside in a typical resting membrane potential? Is it because we're gaining negative ions like chloride, or because we're losing positive ions? Losing positive ions. And what are those positive ions that we're losing? No. Sodium's going to go into the cell. Its higher concentration is outside. There you go, potassium. All right, so one of the factors that creates a resting membrane potential is that there are more leaky potassium channels than there are leaky sodium channels. They're both positive, single charge positive ions. So we're losing more positive ions from the cell via potassium than we are gaining via sodium, because the cell is more permeable to potassium, meaning we have more of those channels. So we have a higher positive ion loss that creates a negative state inside the cell. All right? What's another type of channel that I don't have listed up here?
Sodium, potassium pumps.
Active transport pumps. It could be for different ions. The most common is sodium and potassium. It can be sodium and hydrogen. All right, so we have a sodium-potassium pump. In this case, it's also unequal status. And so with an active transport, remember we're going up the gradient. So which way is sodium being pumped? Out of the cell or into the cell? Out of the cell. So we're pumping sodium out of the cell and potassium into the cell. But for every 3 sodium ions leaving the cell, we're only gaining 2 potassium. So that's a second reason why the inside of the cell is more negative than the outside of the cell. The third doesn't have anything to do with the membrane. It has to do with what's trapped inside the cell, and it's too big to leave to get across the membrane. We have protein molecules inside the cell, enzymes or structural proteins that have a net negative charge. And that adds to the negative nature. So intracellular proteins with a net negative charge. So in our muscle cells and our nerve cells, if nothing else is happening to the cell, these three components or conditions will give us a negative resting membrane potential. Okay? So that's what we have drawn up here.
Can you please explain the third one again, real quick?
Which one?
The third one.
The third one? All right, so proteins are not going to go into or out of the cell. They're made by the cell. Some of them are neutral. They have balanced, a positive ion from the nitrogen and maybe a negative ion on the carboxyl end of the protein. But some of them have more negative charges than they have positive charges, and that contributes to the negative balance within the cell. All right, so on the diagram on the screen, it says identify this event where the arrow is. So the resting membrane potential is flat, and then it ticks upward. Depolarization. So it's more polarized the farther away it gets from zero. So when we get closer to zero, meaning no difference, we're depolarizing. All right? So that event would be most likely due to which of the following? A. Adding more positive ions to the cell. So adding more leaky channels, or opening a ligand or a voltage-gated channel that allows positive ions to come into the cell. So what would cause this? There's two. Most common would be an outflow of potassium. In that case, we're losing positive ions, one of the same steps that's keeping us negative, or adding chloride ions. Well, that's not going to happen with muscle cells. It's something that can happen with nerve cells. Okay, that's what glycine does as a neurotransmitter, is it opens chloride channels, but we're not concerned about that with muscle. All right, so this is the membrane potential. Notice I didn't say resting. Do you see any resting parts of that membrane potential? Do you see any straight line? No. So this is the membrane potential of specialized heart cells that used to be muscle cells, known as autorhythmic cells. This event is ongoing. So let's leave that up there for just a moment, and let's look at the cells that you do understand. Can I erase these three components here? So let's look at a skeletal muscle cell. And let's look at this specific region. Remember what that was called? It's going to interact with the synaptic
cleft.
bouton. So the cleft would be the space. The gutters would be in between. But what do we call that specialized membrane? Postsynaptic membrane, or motor. It's also known as the motor end plate. So on that motor end plate, we have receptors. So let's straighten that out and draw it out here a little bit. And that receptor is a receptor for acetylcholine. All right, so when acetylcholine binds to this receptor, it causes the inside of the cell to become more positive. Okay? It does so in a special, unique way by causing this channel to open. This is an ion channel. So it's not a G protein pathway that we've been looking at. This is directly, the receptor directly changes its shape when the acetylcholine binds to it. By the way, this is the same way acetylcholine works when it binds to a postganglionic neuron. Okay, so it opens an ion channel. And it's always positive sodium ions that come in. So, sodium ions diffuse into the cell. And we're going to see our resting membrane potentials start to become less negative. Depolarization. And over here in our plasma membrane is going to be a voltage-gated cell, receptor. And it will only open if the current or the flow of charged particles going by it is of sufficient strength. If it is, I'm not drawing both activation and inactivation gates, I'm kind of simplifying this. So this is a voltage-gated channel, sodium or potassium, and both of them are going to be effective. So will just one acetylcholine binding to one acetylcholine receptor in the motor end plate allow enough ions in to open the voltage-gated channel? Probably not. So what happens to the change in our membrane? It recovers and comes back to a resting membrane potential. So what do we call this type of a potential? The resting membrane potential was our flat line maintaining a negative state inside the cell. It became slightly more positive and then returned, but not strong enough to open the voltage-gated channels. Graded potential. Subthreshold, yes, but that's a graded potential. So up here is threshold. What am I going to have to do to get my membrane potential positive enough to reach threshold looking at this event right here? I'm going to have to open more channels, okay? So several hundred more ligand-gated channels are going to have to open to create sufficient strength. So I want you to be able to recognize that another form of a potential is a graded potential. And graded potentials are formed using leaky channels, as we'll see in just a moment. Ligand-gated channels. And if we were being thorough, we would say mechanically gated channels as well. Okay, but for our events today, we're going to be looking at leaky channels and ligand-gated channels. Hopefully this is at least familiar. Okay, if you need a refresher course, you can go back earlier in your textbook and with the nerve, depending on whether you have Martini or Marriott, this will be with nerve or with muscle. Okay. Once we reach threshold, and there's another potential for you. And threshold is defined as that current at which the voltage is strong enough to open our voltage-gated channel. What happens when we reach threshold?
Action potential.
Action potential. So we open our voltage-gated channels, and an action potential is formed. What type of channel did I say? Voltage-gated. Okay? So we're going to be comparing the membrane potentials for three different cells. Skeletal muscle cells, which you're already familiar with from Bio 430. autorhythmic cardiac cells and cardiac muscle cells. And you should, for any of those, be able to tell me what type of a potential is being formed and what type of channel is contributing to that potential, all right? It should be review material. If you don't remember it, it should come back relatively quickly. We're going over it. But this is the basic background that you will need to be able to do that and to understand those concepts. Okay? So, this is the current membrane potential changes that would be seen with an electrode inside an autorhythmic cell of the heart, where if you've heard of SA node, that's formed of a whole bunch of autorhythmic cells. Okay? And one outside the muscle cell, which isn't something we do as an everyday test on people. Okay? Requires isolated materials and cells and stuff. But if we were to do that, this is what we would see. No resting membrane. There's something going on all the time. Can I erase what's on the board? Okay, and I'm going to go through this specific sequence. So cardiac autorhythmic cell. And if we were to look at this current, we would see this that's a diagram that you should recognize as the membrane potential of a cardiac autorhythmic cell. So let's look at it based on what we just reviewed. All right? Can you identify depolarization? Okay. Depolarization is occurring here and here. Then we have repolarization and then depolarization again. Okay? So that's the cycling that's occurring over and over in these autorhythmic cells. Now, what's contributing to this? The term autorhythmic means the cell is creating this rhythm itself. This would occur without any nerves releasing neurotransmitters. This is not ligand-induced by neurotransmitters or hormones. If you cut the heart out of a person, okay, so if someone needs a heart transplant, someone has died and they're keeping the heart pumping with machines, even if they were to turn off the machines and take the heart out and put it in a saline condition, all right, that heart could be stimulated to pump and then it would keep pumping. We don't do that when we want to do heart transplants because we don't want to build up the lactic acid and the pH changes that occur. But if we were to pet the frog, that means to destroy brain tissue and then down the spinal cord with a straight needle and open up the heart, open up the thoracic cavity. You could cut the heart, cut the superior vena cava and aorta and inferior vena cava, pulmonary arteries and veins, and take that heart and place, or a turtle heart or whatever, put it in a beaker with the proper salt, and the heart would beat, I've seen them beat for at least 45 minutes without any nerves. Okay? So, heart transplants, when the new heart is placed in the recipient, they don't sew the nerves back together. They obviously have to sew the blood vessels back together, okay, but they don't sew the nerves back together. And the heart will beat on its own due to these cells. So these cells are creating the action potentials from which the electrical current is derived that activates the voltage-gated channels in our cardiac muscle cells. So in a skeletal muscle cell, we wanted it to contract. We had to activate these receptors here. And then that current, if it was strong enough, would activate voltage-gated channels in the cell membrane that would then travel deep into the cell via the transverse tubules, all right, and open up the sarcoplasmic reticulum and so on. With heart, we don't need this. What we need to do is a current strong enough to open up the voltage gated channels directly. And that's what the autorhythmic cells create. So every time an autorhythmic cell has an action potential, that current travels out of the autorhythmic cell via gap junctions to a contractile cell that it's attached to, through that contractile cell to another. Remember the branched cells and the gap junctions between them? And that current spreads throughout the atria and up the ventricles. But this is the origin of it right here. So how do we do that? Would this be an action potential or a graded potential? A graded potential. So we need a strong enough number, a high enough number, a strong enough current, and a current is just a flow of charged particles. So we need a strong enough current to reach threshold. That's obtained in our autorhythmic cells through leaky sodium channels. You've seen minute timers. What's that the second movie, The Night in the Museum, where the guy's trapped inside the hourglass? Okay, so as you visualize those sand granules flowing down to the bottom of the minute timer or the hourglass, visualize the sodium diffusing into the autorhythmic cell. And every sodium ion passing through a leaky channel is going to cause this positive current to increase, purely by leaky sodium ions. So in autorhythmic cells, we have a balance where the cell membrane is more leaky to sodium rather than more leaky to potassium. So we have a gain of sodium. Eventually we reach threshold. So this is a graded potential due to leaky sodium channels. We reach our threshold potential. And that means that this is an action potential. The halo is just showing both sides of the action potential, not a true halo. And normally